So hello students, welcome back to your own channel Vedantu NEET English, and this is Baswaraj sir, your biology master teacher. Now students, this is the second lecture, right? This is the second lecture from the cell cycle and cell division. In the first part, we finished all the mitosis, right? We finished mitosis, what is the cell cycle, all that we finished in the first part. Today is going to be the last and the final part where we'll be dealing with meiosis. Especially the prophase one, the troublesome prophase one is there, no? The troublesome prophase one. That leptotene, zygotene, pachytene, diploidine, dikinesis. All those things we'll be dealing in today's class. And also in today's class, by the end of today's class, we will be solving a lots of PYQs. Now students, let me know in the chat if I'm audible and visible first of all. Yes, if I'm audible and visible. Quickly let me know in the chat if I'm audible and visible also. Now that PYQ reminds me, here I have a question for you. Students, look at this question. After meiosis 1, the resulting daughter cells have the amount of DNA compared to haploid. It looks like a difficult question, but it is actually a very easy question if you know the DNA content. That's right. If you know the DNA content, this particular question is actually very easy. And also, if you look at this question, a numerical. It is a numerical. It may seem difficult at first, but it is very easy. Very easy numerical. In fact, look at this numerical here. A fruit fly numerical. You might think it is from your genetics, but it is not. It is not from genetics. It is from the cell chapter. So students, stay tuned till the very end, because all of these such questions, right? All of such questions we will be dealing in today's class. All such questions will be dealing in today's class. Okay? So let me know in the chat if you're ready for today's class, because it's going to be a blast. And let's have fun, right? Let's have fun and learn meiosis today, right? Let's have fun and let's learn meiosis. Before that, let me zoom this. Let me know if it is clearly visible, first of all, students. Let me know if it's clearly visible to all of you. Yes, it's clearly visible. If it is very clearly visible, quickly a fire in the chat, and also green as in the chat, because we need energy, right? Because we are here from morning 10 o'clock till night 8 o'clock. So unless you show the energy, it is, not very encouraging, right? So let's start the class with meiosis. First line of NCERT. Here we go. Right? Meiosis, right? Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes. First of all, tell me, what is the major difference? What is the major difference between sexual reproduction as well as asexual reproduction? Quickly in the chat, go. Now, all of us know sexual reproduction involves fusion of gametes. Do you see that in the case of asexual reproduction? No. We see the fusion of gametes only in the case of sexual reproduction. Okay, that is fusion of gametes, each with a complete haploid set of chromosome. So what they're trying to tell you in the second line here is that the gametes are gametes. The gametes are haploid in nature. Gametes are haploid in nature. That could be sperm or could be the egg. They are haploid in nature, right? Now, the gametes are formed from specialized diploid cells. That's right. It is formed from a specialized diploid cell. This diploid cell is nothing but the germ cell. They are the germ cells. Now, this germ cells reminds me, can anyone tell me what is the male germ cell called as? Quickly go in the chat. What is the male germ cell called as? That is spermatogonia, right? What about the female germ cell? The female diploid cell, the female germ cell or the diploid cell is oogonia. This oogonia or spermatogonia later on undergoes meiosis to produce the sperm and the egg. Sperm and the egg. That is the germ cell which are diploid in nature. Diploid in nature. Okay. Any doubt there? Any doubt there quickly in the chat? No. This specialized kind of cells division that reduces the chromosome number by half, resulting in the production of haploid daughter cells. Very important line. What does it tell you? This line tells you basically that we have in the case of meiosis, a diploid cell will change its ploidy into haploid. Diploid cell will become haploid here. Okay, and this haploid is nothing but the gametes. Is the gametes. They are the gametes. Okay, they are going to be the gametes. Clear? All of you? Clear? Next one. This kind of division is called as meiosis. Now students, tell Tell me, what about in the case of mitosis? Was there a change in ploidy? True or false? Quickly in the chat. In the case of mitosis, was there a change of ploidy? True or false? Now, meiosis ensures the production of haploid phases in the life cycle of the sexually reproducing organisms. That is the main plant body. So, or the main human body. All of us are humans, right? We are diploid or haploid. We are diploid. So, we have human here. We have human. Let's take a male and a female. We are diploid in nature. We are diploid in nature. We produce gametes. We have sperm and the egg, which are haploid in nature. Again, they'll fuse together to produce the zygote, which is again diploid in nature. So from diploid to haploid to diploid. So in the end, we end up with diploid only. That is called as conservation of ploidy. That is called as conservation of ploidy. If you look at here, it's given here, see. Of sexually reproducing organisms where fertilization restores the diploid phase. So from diploid phase, this is the diploid phase to haploid phase, diploid phase to the haploid phase. Again, we have the diploid phase. This is called as restoring of ploidy. Restoring of ploidy is happening. Easy. Restoring of ploidy. Okay. Students, voice is breaking?
Yes.
A lot? Yes or no? Not hearing, huh?
आवाज आ रही है.
Okay, 1 second, students. 1 second. 1 second. 1 second. Let me get it correct.
See, मैं देख के खुद ही आ गया. How good I am.
Smart. 1 second, students. 1 second. 1 second. We're getting rectified. Not a big deal.
इसको भी निकाल.
हाँ, remote here.
और एक चीज.
हाँ.
वो जो क्लिप है ना.
हाँ. One second. Will it change? Check. Now, sound check, students. Tell me if the voice is breaking now. Tell me.
That's not.
I hope there is no voice breakage now because we changed the wire only now. Full smart, fast people we have here. Okay. Now that is called as conservation of ploidy. Now we come across meiosis during the gametogenesis, right? During gametogenesis, I told you, as the spermatogonia undergoes meiosis to produce the sperm, oogonia undergoes meiosis to produce the egg. That is gametogenesis. In plants as well as in animals, this leads to the formation of haploid gametes. The key feature of the meiosis are follows. Key features are the following. Let's read the key features now. Any doubt now? Tell me. First paragraph, any doubt? Please tell me. First paragraph, any doubt regarding meiosis? Very easy, students. Very easy it is. Very easy. Okay. Now, students, what are the key features? Now, before we go to the key features, let me teach you how many parts meiosis is divided into. How many parts meiosis is divided into? Understand this. The entire meiosis, students, entire meiosis can be broadly classified into two categories. Broadly classified into two categories. Okay. That first one is called as interphase. Okay. Interphase. Then we have the meiosis here. Meiosis. Okay. Meiosis. Now this interphase and meiosis. Now this meiosis is further classified into three categories. Okay. We have meiosis 1 and we have meiosis 2. Meiosis 1 and meiosis 2. Between meiosis 1 and meiosis 2, there is one more phase. Between meiosis 1 and meiosis 2, there is one more phase called as interkinase. Interkinesis or interkinesis. So if you broadly speaking, meiosis divided into interphase and meiosis. Meiosis further classified into meiosis 1, interkinesis, and meiosis 2 as well as and meiosis 2. Any doubt there? Any doubt, students? Please tell me in the chat. There should be no doubt there. Okay. Now, meiosis involves two sequential cycles of nuclear and cell division called as meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. But only a single cycle of DNA replication. What is the meaning of this? What is the meaning of this? A single cycle of DNA replication. Very simple. Very simple. Understand? Now, this particular phase which we have here, that is interphase. Interphase will have G1, S and G2. G1, S and G2. What exactly happens in S, students, tell me? In S phase, all of you know there is DNA replication. There is DNA replication takes place here. DNA replication takes place here. Okay. Now, you need to understand that interphase and interkinesis are almost alike. They're almost alike. But there is one major difference. The major difference between interphase and interkinase is that in the interkinesis there is no DNA replication. There is no DNA replication. Completely absent. So in the entire of your meiosis, the only time where the DNA replication takes place is at the S phase of the interphase. Interkinesis, no. Interkinesis will have other features like your interphase. Like in interphase, we had in the S phase, do you remember? In the S phase, we also had duplication of centrioles in the cytoplasm. All that will happen in interkinase. But there will be no DNA replication. Clear? Friends, clear? Done? Understood that point? That point is very important. That point is going to be very, very important for everyone. That interkinesis, in the interkinesis, there is no DNA replication. No DNA replication. Now, next point. Understand? So there is only a single cycle of DNA replication. This is the only part where the DNA replication takes place. Okay. Meiosis 1 is initiated after the parental chromosomes have replicated. That is, after interphase. After interphase, we have the meiosis 1. After interphase, we have the meiosis 1 here. After interphase, we have the meiosis 1, where in the S phase there is DNA replication. In the S phase there is DNA replication. See here, to produce the identical sister chromatids at the S phase. Now, meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids of homologous chromosomes. That is basically what is happening in meiosis 1. In meiosis 1, there is something called as leptotene, zygote, pachytene. All that we will discuss, where we are pairing of homologous chromosomes, crossing over. We'll get to that. Okay, this point we'll get to that. 4 haploid cells are produced at the end of meiosis 2. So, at the end of meiosis 2, students, at the end of meiosis 2, 4 haploid cells are obtained. 4 haploid cells are obtained at the end of meiosis 2. That's it. That's it. Okay, meiosis 2. Now, next question. Now, meiotic events can be grouped under the following phases. Now, students, you need to understand one thing here. That is, this meiosis 1 right here and meiosis 2 right here, it can be further divided into the same PMAT. All of you remember, right? PMAT. That is prophase, metaphase, anaphase, as well as telophase. The same PMAT. So, but here we have something called as, in meiosis 1 we have prophase 1, metaphase 1, anaphase 1, telophase 1. In your meiosis 2 we have prophase 2, metaphase 2, anaphase 2 and telophase 2. That's all. Any doubt there? Quickly tell me. Students, if you're able to understand, quickly smash that like button, all of you. Smash that like button. If you're watching it recorded also, smash that like button. Right? Madhumita is pointing it out. Yes, PMAT. Now, students, with that basic information about your meiosis starting, the basic information like we have PMAT, all of those information. We have We know interphase, meiosis, meiosis 1, meiosis 2, interkinase. With this basic information, can we start now prophase 1? Can we now start with prophase 1? Quickly, green hearts in the chat if you're ready for it. Quickly in the chat. Green hearts in the chat if you're ready for it. Quickly, all of you. And say hi to my pink bottle. All of you drink some water. Keep drinking water. Drinking water is very essential. So keep drinking water always. Drink more water. Okay. Now, let's start with your meiosis 1. Meiosis 1, that is prophase 1. Prophase 1. Now, you need to understand what exactly happens in your prophase 1. In the prophase 1. Prophase 1. Now, students, you need to know that this prophase 1 can be further divided into multiple different levels. For example, this prophase 1 right here, can be further divided into multiple types. That is, let zebra play during the day. Let zebra play during the day. What is this, sir? What is the meaning of this? Understand. One second, let me erase this, let me write it up. Let zebra play during the day. What is the meaning of this? This is prophase 1. Prophase 1. From here, students. Understand, from here we are starting now. Prophase 1, we are starting this one, okay? What is this let zebra play during the day? Here, understand, this L stands for, the L stands for leptotene. The L stands for leptotene. Leptotene. What is Z? The Z stands for zygotine. Z stands for zygotine. What is this second P? P stands for pacatine. C stands for pacatine. Now what is this D? D stands for diplotene. Okay. Diplotene. Now what is this final D, students? Then what is the final D? The final D is nothing but the diakinesis. 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 So let zebra play during the day is basically telling you, the different parts, the different stages of prophase 1. Different stages of prophase 1 is telling you, let zebra play during the day. That is leptotene, zygotine, pacatine, diplotene and diakinesis, as well as diakinesis, the final D. Students, will you remember this? And the same order. Students, don't mix up the order now. The same order should be there. The same order. Because there will be questions. And there has been questions on the sequence of the prophase 1. Sequence of prophase 1. Leptotene, zygotine, pacatine, diplotene and diakinesis. 5 of them. Okay. Leptotene, zygotine, pacatine, diplotene and diakinesis. Now, I might ask you a question. On what basis are we dividing that? On what basis do we have leptotene, zygotine, pacatine, diplotene and diakinesis? What is the foundation and what is the base around it? It's very simple. It is the chromosomal behavior. So can you give me a proof? Where is it written? Right here. See this. That is prophase of mitosis. The prophase of your first meiotic division is typically longer than the more complex compared to the prophase of mitosis. Obviously, prophase 1 is much more complex compared to mitosis. Understand the point? It has been further subdivided into following 5 phases. What is the basis here? Based on the chromosomal behavior. You might skip this point, but it is an AR question. Question is very simple. Assertion. Prophase 1 can be divided into leptotene, zygotine, pacatine, diplotene, diakinesis. Reason. It is divided based on the chromosomal behavior. So what would be the answer? Option number A, assertion is right, reason is right. Reason is the correct explanation of assertion. Do you understand? This is how question can be asked. If you do not know every single line here, you're bound to go a mistake. You're bound to make a mistake. Okay? Clear? Understood? Chromosomal behavior. Chromosomal behavior. Now, next one. Now we'll understand leptotene now. Let's start with leptotene. Okay, let's start with leptotin first now. Okay, during leptotin stage, the chromosome becomes gradually visible under the light microscope. Students, all of you, see this. Varun, Tanuja, Jansli, Savita, all of you see this. Very important point here. Very important point. This one line here, this one line here is actually a PYQ. This question right here has been asked for around 3 to 4 times. What exactly happens in leptotin? What exactly happens in leptotin? So what is the first point? The chromosome becomes gradually visible in microscope. What is the meaning of that? Condensation of chromatin is happening. What is happening? Condensation of chromatin is happening here. Condensation of chromatin. Condensation of chromatin. Condensation of chromatin is happening here. That is why under leptotin they are visible. They are visible. Okay. So during leptotin stage, chromosome become gradually visible under the light microscope. PYQ question. The compaction of chromosome continues through the leptotin. That's all is leptotin. That's all is leptotin. So if they ask you a question around leptotin, if they can ask you a question around leptotin, only thing they'll ask you is which is the first stage in meiosis in the prophase 1? Answer should be the first stage in prophase 1 is leptotin. What exactly happens in leptotin? Condensation of chromatin. And because of the condensation of chromatin, students, because of condensation of chromatin, the chromosome becomes gradually visible under light microscope. 3 points, that's it. That's it. 3 points for leptotin. Done. Done. Now students, let's go to the next stage. The second stage is called as zygotin. The second stage is called as zygotin. So this is your leptotin. So can you see? Under leptotin, what is happening? The chromosomes are becoming gradually visible under microscope. The homologous chromosomes. Now understand zygotin. Now understand zygotin. Clear? Clear, right? All of you? Now apart from that extra information, if I tell you it's called as bouquet stage, there is something called as formation of asters, all that happen in leptotin only, but not given in NCERT. They will never ask you out of NCERT out of here. Understand that. Now let's go to zygotin. Let's go to zygotin. What exactly happens in zygotin, friends? Now understand zygotin here. Understand zygotin. This is one chromosome. This is one chromosome right here. This is one chromosome. We have one more chromosome which looks just like the first chromosome. Same to same. Twins there. Twins. Okay. Right. For example, we have Paul and we have Meghna and let's say Riyadh. Okay. Both of them. Okay. Both of them are twins here. Twins. Now I can call these twins as, there's a term for it. When the two chromosomes are look alike. Okay. I can call them homologous chromosomes. Homologous chromosomes. They're called as homologous chromosomes. Okay. I can call them homologous chromosomes. Homologous chromosomes which look alike. Okay. They're called as homologous chromosomes. Now, how many of you played any games in the classroom? Tell me. In group teams, always in the class, you have one best friend, right? This is like a best friend. All of you, anytime there is a group project, you will make a small group of 2, right? Group project, if you make a small group of 2, you always make a bond with your friend, best friend, both of you become a team, right? Here also we have something called as pairing of chromosomes, like how you pair up with your best friend. Here also what is happening? One chromosome is pairing with the other chromosome. One chromosome is pairing with the other chromosome. This is called as pairing of homologous chromosome. And the process by which the two chromosomes are coming together, the process is called as your, where is it written? The process is called as synapses. The process is called as synapses. When the two homologous chromosomes, students, when the two homologous chromosomes, they come together, that is called as synapses. That is called as synapses. Is that point clear to all of you? What is synapses? What is synapses? When the two homologous chromosomes are associating with each other, the two homologous chromosomes are coming together, the process is called as synapses. Done. Now students, you need to understand few points here. They are called as sister chromatids. They are called as sister chromatids. Sister chromatids. Okay, they are called as sister chromatids. Now students, between the two homologous chromosomes, there is some form of complex formation here. Can you see? Between the two homologous chromosomes, there is some form of complex formation. Complex formation between the two homologous chromosomes. The complex which is formed between two homologous chromosomes is called as synaptonemal complex. It's called as synaptonemal complex. Synaptonemal complex formation. Synaptonemal complex formation. Well, understood. So what is happening here in the case of zygotine, students? In the case of zygotine, what do we have? We have formation of synapses. What is synapses? When the two homologous chromosomes are coming together, that is called as synapses. And between the two homologous chromosomes, there is some form of complex. The complex is called as synaptonemal complex. It is called as synaptonemal complex. Understood, all of you? Tell me. Understood? That is called as synaptonemal complex formation. See here. During the stage of chromosome starts pairing together, and this process of association, I told you, the process of two homologous coming together, that is called as synapses. That is called as synapses. Such paired chromosomes are called as homologous chromosomes. Electron micrograph of this stage indicates that chromosome synapses is accompanied by formation of a complex. The complex is called as what? Synaptonemal complex. It is called as synaptonemal complex. Synaptonemal complex is formed between the two homologous chromosomes. Done? Yes? Thumbs up or down? Up or down? Quickly in the chat. Now, the complex formed by the pair of synapsed homologous chromosome, there is another name for this. The other name here which is given here is called as bivalent or tetrad. Bivalent or tetrad. This stage right here is called as bivalent. It's called as bivalent, and it can also be called as your tetrad stage. It can also be called as what? Tetrad stage. And let me tell you, the meaning of bivalent and tetrad both are same to same. This right here is same. Now understand this point. The reason why they are called as bivalent, because of the two chromosomes. Because of the two chromosomes. Can you see? One centromere here, second centromere here. That is why they are called as bivalent. Now why are we calling them tetrad? We are calling them tetrad because, tell me, how many chromatids do you see here? 1, 2, 3 and 4. Do you see 4 tetrads here? Yes or no? 4 tetrads here. So that's why it's called as tetrad stage. Tetrad stage. 4 chromatids. 4 chromatids. 4 chromatids. So this stage right here, when the two homologous chromosomes are coming together, there is a complex formation, synaptonemal complex, and this stage is called as bivalent, or it can be called as tetrad stage. Bivalent or tetrad stage. Now you need to understand one more point here which can be asked multiple times, has been asked this question. This bivalent or tetrad stage, where does the formation happens? Question will be very simple. Formation of bivalent or tetrad happens in which stage of prophase 1? Leptotin, zygotin, pachytin, diplotin. Tell me the answer. Zygotin is the answer. Zygotin formation happens. But sometimes they can ask you a question. In which stage of prophase 1 the bivalent or tetrad stage is clearly visible? Now there you cannot tell zygotin, because the clearly visible part is happening in the next stage, that is pachytin. That is the pachytin. See here. However, these are more clearly visible at the next stage. So this bivalent tetrad stage, formation is happening in zygotine. Understand the concept. Formation is happening in the zygotine, but it is the clear visibility part, right? The more clear visibility part is in the next stage, that is the Pacatine stage. That is the Pacatine stage. Clear? Pacatine, not leptotine, Madhumita. Leptotine was the first stage. Okay, first stage was leptotine, gone. Pacatine. Understood? See here. Now one more point which has been an AR question, which can be asked as a AR question here, right here. The first two stages, that is leptotine and zygotine. Leptotine and zygotine, the first two stages of the prophase 1 are relatively short-lived. That is, leptotine and zygotine are short-lived compared to the next stages, that is Pacatine. That is next Pacatine. So if they ask you a question, which stage is the shortest stage? They'll give you options: leptotine, zygotine, Pacatine, both A and B. What is the answer? Tell me now. Which are the shortest stages? Leptotine, zygotine, Pacatine, both A and B. Answer should be both A and B. Understood? You can get a question like that. You can get a question like that. Okay. Now, next we go to Pacatine now. Next we go to Pacatine. So this is the homologous chromosomes coming together. That is called a synapsis. Now, what is happening between? Between them there is a complex formation. There is a complex formation. And the complex is called as what? The complex is called as synaptonemal complex. Synaptonemal complex. Students, if there is no doubt in zygotine, green as in the chat, quickly go. That is your zygotine. Synaptonemal complex formation. That is the main goal of zygotine. Okay. Now we come to Pacatine now. Now we come to Pacatine. Very easy again. Very easy. Now we come to Pacatine. Okay. Now we come to Pacatine. Understand. What exactly is happening in the Pacatine? What exactly is happening in the Pacatine? Understand now. Understand. What exactly is happening in the Pacatine? Okay. Now we had chromosomes here, right? Let me draw the chromosomes. Let me draw a different color now for you to understand better. Let's take two different colors and understand. What exactly happens in Pacatine? This is one chromosome here. One chromosome. And we have the second chromosome here. We have the second chromosome here. Okay. They are the two homologous chromosomes. They are the two homologous chromosomes. Between the two homologous chromosomes, there is synaptonemal complex. There is synaptonemal complex between the two chromosomes. Now there is something called as crossing over. There is something called as crossing over. Students, I told you before, these two are called as what? They are called as sister chromatids. They are called as sister chromatids together. What about these two? This one blue color, one pink color. These two are called as non-sister. They are called as non-sister chromatid. They are called as non-sister chromatid. Non-sister chromatid. Okay, non-sister chromatid. Now understand the next point. What exactly is happening? What is crossing over? Crossing over is nothing but. Crossing over is nothing but. Right? Crossing over is nothing but exchange of genetic material. What is happening here? Exchange of genetic material. Exchange of genetic material. Exchange of genetic material between non-sister chromatids. Between non-sister chromatids of homologous chromosomes. Of homologous chromosomes. Between homologous chromosomes. So are they homologous chromosomes? Yes, they are homologous chromosomes. Now what is happening? Exchange of genetic material. Exchange of genetic material between non-sister chromatids. This is the one between, can I see? I'll show you right here. The exchange of genetic material will happen between this one and this one. They are non-sister chromatids. They are non-sister chromatids. Okay, understand now what is exactly happening. Okay, this is here. Can you see the difference now? What exactly is happening? They are crossing over now. There is crossing over. There is crossing over. Okay. Now, after the crossing over takes place, what is the result of crossing over? What is the result of crossing over now? See the result of crossing over. There will be exchange of genetic material. This is one. Here we'll have the pink color one. Yes. It's very simple to understand. Do you see that? What exactly happened now? First we had the two homologous chromosomes, genetic material intact. What happened now? Between non-sister chromatids there was a crossing over took place. Now after the crossing over, whatever strand was here, it came here and this went there. This is called as recombinant. It's called as recombinant now. It's called as recombinant now. It's called as recombinant. Understand with the help of diagrams now. Okay. Now let's read what is given in NCERT now. Let's give what is given in NCERT. Okay. During this stage, the 4 chromatids of each bivalent. This is a bivalent, right? This is a bivalent or it is a tetrad. Bivalent or tetrad. Bivalent or we can call it tetrad. Tetrad. Bivalent or tetrad. Right? Bivalent or tetrad. Chromosomes becomes distinct and clearly appear as tetrads. I told you. Clear appearance is happening in the pachytene. Formation, zygotine. Tetrad formation. Okay. This stage is characterized by the appearance of recombination nodules. First main point here about your pachytene is that apart from crossing over, the other main part is formation of recombination nodules. Recombination nodules. Formation of recombination nodules. Very important point. Formation of recombination nodules. Right. Now, the site at which crossing over occurs between the non-sister chromatids of the homologous chromosomes. So, where do you find recombination nodules? At the site of crossing over. If this is the site of crossing over, here we will see the recombination nodules. We will see the recombination nodules at the site of crossing over. Here we have recombination nodules. Recombination nodules at the site of crossing over. Between them we have the synaptonemal complex still. The synaptonemal complex is still there. Between them, the synaptonemal complex is still there. Remember that. Synaptonemal complex is still there. Okay. Now understand. Understood? So crossing over is happening. Crossing over is happening between non-sister chromatids of homologous chromosomes. This is homologous chromosomes, non-sister chromatids. Okay. Crossing over is the exchange of genetic material between the two homologous chromosomes. Crossing over is also an enzyme mediated process. So understand, the entire thing is an enzyme mediated process and the enzyme which is involved is called as recombinase. That is very important. All of you mark that. All of you mark it very well in your book. That is, the enzyme involved is called as recombinase enzyme. Recombinase enzyme. Mark that point. So, pachytene, what is happening? In the pachytene, there is something called as crossing over, recombination nodules. Second point. The third point is going to be recombinase. Recombinase is going to be the third point. Three main points of pachytene. Okay. Crossing over leads to the recombination of genetic material of the two chromosomes, which results in the recombinant. Okay. Now, recombination between the homologous chromosome is completed by the end of pachytene. It is over by the end of pachytene, leaving the chromosomes linked at a site of crossing over. Now, where are they linked? They are linked only at the site of crossing over. That is the end of pachytene. So, three main points about pachytene is exchange of genetic material, that is going to be a crossing over, formation of recombination nodules, recombinase enzyme. and this bivalent tetrad stage is better visible at Pacatine. Recombination is over at the end of Pacatine. Every single point is a question. I told you 4 or 5 points now, right? Every single point is a PYQ. I'm not even kidding. Every single point is a PYQ here. Every single point is a PYQ. That is Pacatine. Okay, that is going to be Pacatine. What is zygotine, students? In zygotine, what did we learn? In the case of zygotine, we learned about what did we learn in zygotine? In the zygotine, what is happening? The two homologous chromosomes are coming together. That is called as synapsis. Between the two homologous chromosomes, there is a complex formation. That is called as synaptonemal complex formation. That's all. That's all. And here we have a stage called as bivalent or tetrad stage. Bivalent is called as because of 2 chromosomes. Tetrad because of 4 chromatids. Now we will go to the next stage now. See, this is the crossing over. Now students, the next one is diplotin. The next one is called as diplotin now. Diplotin, next stage. Very easy again, diplotin. Now students, here, where is the chromosomes? Where are the chromosomes? This is one chromosome. This is the one chromosome now. This is the other chromosome, right? This is the second chromosome. The 2 chromosomes are here. There are 2 chromosomes here. Between the 2 chromosomes, there is something called as synaptonemal complex. There was synaptonemal complex. So what exactly happens in diplotin, students? What exactly happens in diplotin? What exactly happens in your diplotin? What exactly happens in diplotin? In the case of diplotin, students, understand this point. In the case of diplotin, what is happening is the synaptonemal complex. The synaptonemal complex is completely removed. Synaptonemal complex is completely being removed. So which was looking like this, your chromosome which was looking like this will end up looking, I'm not doing the color again now, students, understand. So synaptonemal complex which is looking like this, the synaptonemal complex will disappear. This will disappear. It will completely disappear. Okay. Now this will completely disappear, but except at one point. The entire synaptonemal complex will disappear except at one point, that is this X mark. Can you see this X mark here? Can you see, students? The entire synaptonemal complex will disappear except at one point. That one point, this X mark is called as chiasmata. This X mark is called as chiasmata. Read the lines of NCERT now. See what is given here. The beginning of diplotin is recognized by the dissolution of the synaptonemal complex. Told you. So by the end of diplotin, by starting of diplotin, not end, starting of diplotin is what? The synaptonemal complex is disappearing. It is completely disappearing. Okay, understand. And the tendency of the recombined homologous chromosomes of the bivalent to separate from each other. Can you see? Now what is happening here? The 2 homologous chromosomes are separating from each other. The 2 homologous chromosomes are separating from each other except at one point. Except at the site of crossover, this X-shaped structure are called as chiasmata. It's called as chiasmata. This right here. This right here, can you see this part? This X-shaped mark. This X-shaped mark at the crossing over. This X-shaped mark at the crossing over. The X mark. The X mark at crossing over is called as chiasmata. It's called as chiasmata. It's called as chiasmata. The X-shaped mark is called as chiasmata. So if someone asks you, students, what comes to your mind when you think of chiasmata? You should blindly tell me, sir, it is diplotin. Chiasmata, diplotin. Dissolution of synaptonemal complex, or disappearing of synaptonemal complex, diplotin. Understanding? Diplotin. Now there is one line here which is a PYQ many times. In oocyte of some vertebrates, example egg, in the case of female, diplotin can last for months or several years. Diplotin stage. So a particular cell can be arrested at diplotin stage. The egg cell. The egg cell in the case of female, the secondary oocyte gets arrested, right? The secondary oocyte gets arrested. So the primary oocyte gets arrested for a long time in the diplotin stage. And this is a PYQ. This right here is a PYQ. That is the diplotin stage. Now what is happening? This is a diplotin stage. Now what is happening in the last stage, that is diakinesis. Now this is the two chromosomes, this is the X mark. The X mark is called as what? The X mark is called as chiasmata. All of you know that. Now what exactly is happening at the end of in the diakinesis? This X-shaped mark will also come apart. That is called as termination of chiasmata. This is chiasmata, right? Now what is happening in diakinesis is that this X-shaped mark is also coming out. That is called as termination of chiasmata. The final stage of prophase 1 is called as diakinesis. This is marked by the termination of chiasmata. Very important point. So chiasmata which was formed in the stage of your diplotin chiasmata, the dissolution of chiasmata takes place in your diakinesis. So by the end of diakinesis, the entire chiasmata is terminating. During this phase, the chromosomes are fully condensed. That is the main goal of prophase, right? The complete condensation of chromosomes. And meiotic spindle is assembled. That is spindle fibers is coming up to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears. Remember, in the case of your prophase also, nucleolus is disappearing. Yes, nucleolus disappearing, and nuclear envelope also breaks down. So students, remember, nuclear envelope breaking down is given towards the end of your prophase. That point is very clear. Remember, this is happening. The breaking of nuclear envelope is happening at the end of prophase. So can I call it technically late prophase? Technically the late prophase at the end. Also breaks down. Diakinesis represents transition into metaphase. That's it. Prophase 1, that is meiosis 1 is over. I'll recap one more time with every single main point. Just understand now. Every single point I'll recap one more time. Just follow it one by one. Just follow it. Let's from starting. The main points I'll tell you. Main points now. Main points. Your entire meiosis 1. The entire meiosis 1 is divided into. The entire meiosis divided into interphase and meiosis. Interphase divided into G1 as G2. Meiosis divided into meiosis 1 and meiosis 2. Between meiosis 1 and meiosis 2, we have interkinase where there is no DNA replication. Now, meiosis 1 can be further divided. Meiosis 1 can be further divided into multiple parts. Right? Meiosis 1 can be further divided into prophase, metaphase. Meiosis 1 can be further divided into, there's a slide. Meiosis 1 can be divided into prophase 1, metaphase 1, anaphase 1 and telophase 1. 1. Now this prophase 1 can be further divided. Into leptotin, zygotin, pachytin, diplotin and diakinesis. Leptotin, zygotin, pachytin, diplotin, diakinesis. The entire prophase 1. Okay. What exactly happens in leptotin? What exactly happens in the case of leptotin? Now students, remember, the basis of classification is chromosomal behavior. Now in the case of leptotin, what is happening? In the case of leptotin, condensation of chromatin. And chromosome become gradually visible under light microscope. See here. During leptotin stage, chromosomes become gradually visible under light microscope. Main point. Now zygotin. What exactly happens in zygotin? In zygotin, the two homologous chromosomes are coming together. The process by which, the process is called as what? The process is called as synapses. The two homologous chromosomes coming together is called as synapses. It's called as synapses. Okay. So we have synapses. In the synapses, there is a complex formation. The complex is called as synaptonemal complex. The entire structure is called as bivalent because of two chromosomes, or it can be called as tetrad. Tetrad, why? Because of 4 chromatid. That's all is the main points in the case of zygotine. Formation of bivalent or tetrad. Now tell me in the chat, when is it more clearly visible? This bivalent or tetrad is more clearly visible in the Pachytene stage. That is the next stage. That is Pachytene stage. Here, Pachytene stage, more clearly visible. Now, what are the main points about Pachytene? First of all, first point is bivalent or tetrad are clearly visible. First point. Second point is going to be crossing over. Crossing over between what? Crossing over between non-sister chromatid. Crossing over between non-sister chromatids of homologous chromosomes. Crossing over. At the site of crossing over, what do we have? Recombination nodule formation. Yes, that is the recombination nodules. And then finally we have the recombinant. Is it enzyme mediated? The entire process, is it enzyme mediated? Yes, it is enzyme mediated. Can you see? Recombinase enzyme is there. Recombinase enzyme is there. That is Pachytene. Now, next we have diplotin. What happens in diplotin? D for disappear. Diplotin disappears. What is happening here? The entire synaptonemal complex is disappearing, except at one point. That one point is called as X mark, the crossing over point. The X mark or crossing over point is also called as chiasmata. It is called as chiasmata. That is your diplotin stage. The final disappearing. What is happening final? In the diakinesis, there is termination of chiasmata. Termination of chiasmata. Can you see here? That is going to be your diakinesis. Termination of chiasmata. In diakinesis, there is entire nuclear membrane is disappearing. Nucleolus is disappearing. Right? Entire thing is happening here. Homologous chromosomes are completely condensed. Complete condensation is happening. And that's the end of prophase 1. End of prophase 1. Done. Full. One time. Entire revision. I believe there should be no doubt here. Now students, we have metaphase now. Now we have metaphase 1. What exactly happens in metaphase 1? Now let me give you a quick recap. What is to be in the metaphase of your mitosis? Metaphase of mitosis. See this. This is your metaphase of mitosis. In metaphase of mitosis, all the chromosomes are aligned on the equatorial plate. All the chromosomes are aligned on the equatorial plate. Yes. But tell me in the chat, how many metaphasic plate did you see in the case of your mitosis? Only one. There is only one plate here. Can you see? There is only one plate in the case of mitosis. But in the case of meiosis, metaphase 1, we have two different plates. For example, students, let's go back here. I'll show you. In the case of metaphase, of your prophase 1, of the meiosis 1, understand this. In the case of metaphase 1, there are two. Can you see? There are two different plates. And at the plate, what do we have? At the plate, we have the homologous chromosomes, the recombinant ones. Let me draw only one of you so you can remember it. Can you see this? Others I will draw as such. The two homologous chromosomes, remember? Others I will draw as such. Be easy to remember. The crossing over has taken place now, remember. The crossing over has taken place. Because they are present in pair, right? Because they are present in pair. Understand this one key point here. Understand this one. this one, each one of them. How does it look like? Students, doesn't it look like the same chromosome which we had here? Doesn't it look like the same chromosome which we had here? The two chromosomes? Doesn't it look like the two chromosomes which we had here? So this two chromosomes, one and this one, each one of them are separating, right? Each one of them are separating at the end of diakinesis. So after separation, they are present together. So this is actually a homologous chromosomes. So both the homologous chromosomes will align on the equatorial plate, also called as metaphasic plate. But here we have two of them, not one. So we have two metaphasic plate here, metaphasic plate. One more point you should understand here is, here we will have spindle fibers. Obviously, spindle fibers are attaching. Now, the difference between mitosis and meiosis again. In the case of meiosis, one spindle fiber will attach to only one kinetochore of one chromosome. Can you see? One spindle fiber is attaching to the only one chromosome of one chromatid. But in the case of mitosis, how is it in the case of mitosis, students? In the case of mitosis, what did we have? In the case of mitosis, one chromosome. In the case of mitosis, we had one chromosome had two spindle fibers. Can you see? One chromosome, this is mitosis. In the case of mitosis, one chromosome had two spindle fibers attaching. Can you see? One from here, one from here. But in the case of meiosis, one spindle fiber is attaching to only one chromatid, only one kinetochore. Other kinetochore is free here. Other one is free. That point to be noted. Okay. And it is attaching where? It is attaching to the kinetochore. Always remember that. Here we have a disc-shaped structure. Where it attaches. We have a kinetochore here. Kinetochore are present. Okay. Very easy to understand. The bivalent chromosomes align on the equatorial plate. The microtubules from the opposite poles. The spindle fibers. They are the microtubules. Also called as spindle fibers. The opposite poles of the spindle attach to the kinetochore of the homologous chromosomes. They are attaching to the kinetochore of homologous chromosomes. Then that is metaphase. See here, metaphasic plate. Now what is happening in anaphase now? What exactly is happening in the case of anaphase? What is happening in anaphase now? Understand very clearly. In the case of your mitosis, did we learn that this is one chromosome, right? And in the case of mitosis, the chromosome itself split into half. That is splitting of centromere. Where in the case of mitosis, the centromere split and the two chromosomes were pulled apart. Yes. But in the case of anaphase of meiosis 1, that is not the case. In fact, in the case of anaphase 1 of meiosis 1, there is separation of homologous chromosomes. Students, if you go back to this diagram, this one set of homologous chromosomes, set number 1, set number 2. This is one homologous chromosomes, this is the second. The entire thing is homologous chromosomes. This one set will be pulled to this side. This one set will be pulled to the other side. Basically, the two homologous chromosomes are separating. Where? Anaphase 1. Remember that. Now tell me in the chat, simple true or false question. In anaphase 1, the centromere is splitting. True or false? In anaphase 1, the centromere is splitting. True or false? Exactly, it is false. False. Why false? Because centromere is not splitting. the entire homologous chromosomes are being separated out. Entire homologous chromosomes are being separated out. Where? In the anaphase 1. In the anaphase 1. Homologous chromosomes separate while the sister chromatids remain associated at the centromere. So centromere will be present. Centromere will be intact. Only the homologous chromosomes separating out. That's it. That's it. That is your anaphase. Anaphase. Now, then what is happening in telophase? What is happening in the case of telophase? Very simple. Telophase 1 is complete the opposite of prophase 1 again. The nuclear membrane and nucleolus is reappearing. Yes. Cytokinesis follows this and is called as dyad of cells. This is called as dyad of cells. Dyad of cells here. Dyad of cells. Because cytokinesis is happening, we are getting dyad of cells now. Yes. Although in many cases chromosomes do not undergo some dispersion, they do not reach the extreme ends and state of the interface nucleus. Sometimes there is something called as failure of separation of chromosomes, non-disjunction. Okay. The stage between the two mitotic division is called as interkinase. See, it's coming up, interkinase, and is generally short-lived. So it is very short-lived. Interkinase is very short-lived, while interphase is very long. Interphase is very long. Interkinase is very short-lived. There is no replication of DNA. Told you starting, no replication of DNA. Interkinase is followed by prophase 2. So we are prophase 2. So what did we finish, students? Now we finished basically. Now prophase 1. Prophase 1 is over, where we had leptotene, zygotene, pachytene, diplotene and diakinases. Then we did metaphase 1. Then we did metaphase 1, anaphase 1, anaphase 1, telophase 1, telophase 1. After this we had interkinase or interkinases. Interkinases. After that again we have the PMAT. Again we have the PMAT. That is prophase 2, metaphase 2, anaphase 2 and telophase 2. We are done with till here. Till here we are done. Till here we are done. Following? Following? Done. So this is your entire thing is called as your meiosis 1. Meiosis 1. This is your meiosis 2. Meiosis 2. Meiosis 2. So if they ask you a question, when does the interkinase takes place? Interkinases takes place after anaphase and before prophase. Before prophase. Understand that. Easy. Easy. Such easy concepts. Now, students, now I'll tell you one tricky thing. How many of you will agree to this? Let me see. How many of you will agree to this? All of you remember the N condition and C condition which we did in metaphase? N condition, C condition. What if I tell you? What if I tell you the N condition here is N condition? The N condition here is N2C. N2C. The N condition here is N2C. 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 That is with two chromatids. With two chromatids. Here also we have two chromatids. Two chromatids. Tell me how many of you understood this? How many of you understood this? N2C. N2C. How does it happen? N2C. Let's see how many of you can tell me. This is called as a dyad of cells. This is called as dyad of cells. If you do not understand, don't worry. End of the video, I'll tell you. End of the video, after meiosis, after the entire meiosis 2, we will do DNA content. Don't worry about that. Okay. It's called as N2C. I'll get to that. Okay. Now, with that, can we start with Can we start your meiosis 2 now? Meiosis 2. Okay. Now remember, students, meiosis 2 is just like mitosis. This meiosis 2 is just like mitosis. So can I call meiosis 2 as a equational division? equational division. Yes, just like mitosis is called as equational division. So what is happening in prophase 2? Prophase 2 is initiated by immediate after cytokinesis. Okay, usually before the chromosomes are fully elongated. In contrast to meiosis 1, meiosis 2 resembles the normal mitosis. It is just the normal mitosis. Okay, normal mitosis. Okay, now why I am telling Rishmita, why I am telling 2 chromatids is because there is no splitting of chromosomes, so splitting of centromere. There is no splitting of centromere, right? No splitting of centromere. So 2 chromatids. I'll tell you why. We'll get to that. Okay. The nuclear membrane disappears. See, all this is again mitosis only. Disappears by the end of prophase 2 and chromosome becomes again compact. What is metaphase 2? Everyone aligned at the equatorial plate. At this stage, the chromosome align at the equator and microtubules form the opposite poles of the spindle gets attached to the kinetochore of the sister chromatids. Again, similar to your anaphase, splitting of centromere happens. Splitting of centromere. It begins with the simultaneous splitting of the centromere. Okay, at each chromosome, which is holding the sister chromatids together. Basically, splitting of sister chromatids happening here. Okay, allowing them to move towards the opposite poles, shortening the microtubules attached to the kinetochore, just like a mitosis. Again, same to same. Telophase 2. The meiosis ends with the telophase 2, in which the 2 groups of chromosomes once again gets enclosed by a nuclear envelope. That is opposite of prophase. Nuclear envelope is coming back again. And cytokinesis followed by the resulting in the formation of tetrad of cells. 4 haploid cells at the end of meiosis 2. So what exactly happened here, students? What exactly happened here? First we had one diploid cell. This diploid cell underwent meiosis 1. It underwent meiosis 1. In meiosis what happens? In meiosis 1 there is change in ploidy, 2n to n. So we have 2 cells now. Both are haploid in nature. They are called as a dyad of cells. Now what will happen? Each cell will undergo meiosis 2 now. They undergo meiosis 2. At the end of meiosis 2, now tell me, I told you meiosis 2 is just like mitosis. Can there be a change in ploidy? Can this n become n by 2? No. There is no more possibility. So this is meiosis 2 is just like mitosis. So there is only a simple division. N. This is what is happening here. A diploid cell is giving rise to 4 haploid cells. A spermatogonia is giving rise to 4 haploid gametes, 4 haploid sperms. Same thing is happening here. Okay, Now students, this is your anaphase. This is your prophase 2, metaphase 2, anaphase 2, that is splitting of centromeres. Can you see centromeres being split apart? And finally we have the telophase 2. Now friends, I'll tell you one more thing. Here they are called as NC. The cell, the number of the ploidy and DNA content is NC here. How? Tell me how many of you understood this? Why is it called as NC here now? Not 2n2c, not 4n4c. Why are we just calling it NC now? NC, not Hansi, NC. How many of you can tell that? Why do we call them just NC? Here we had 2 and 2c. This is what? This is at the end of telophase 1. Telophase 1. And this is telophase 2. This is NC. How many of you understood this? Tell me. Do you want me to explain that? How many of you understood? How many of you want me to explain? How are we getting NC? How are we getting NC? Want to understand? How are we getting NC and how are we getting 2NC. Understood? Or should I explain? I'll explain what is there. Let's understand DNA content now. If you don't understand, can I teach you DNA content now? So this is the entire prophase. Can you see? Prophase 1, 4 haploid gametes. Diagram is very easy to understand. Before I teach you that, let's do significance. Very simple significance. What is the significance of meiosis? Meiosis is mechanism by which conservation of specific chromosome number of each species is achieved across generation in sexually reproducing organisms. That is 2n to n again back to 2n. That's what they're telling. 2n germ cell, haploid gametes, zygote diploid. This is a zygote diploid. I explained this in the starting of the class. First point. Even though the process per cell paradoxically results in the reduction of chromosomes, yes, by half, it also increases the first one. It also increases the genetic variability in the population of organism. Why genetic variability? Because of crossing over. Okay, from one generation to the next one. Variations are very important for evolution. So if you want to tell, if there was no meiosis, will there be evolution? Tell me, if there was no meiosis, will there be evolution? No. So if you want evolution to happen, there should be variation. And how are the variations created? Because of crossing over. Okay. Now students, let's start with DNA content now. Let's start with DNA content. Ready? Tell me. Ready? Let me check how many of you have marked the attendance. Students, quickly mark your attendance first of all. What is this? What is this sadness? What is sadness? Sumidas, which one you want me to explain again? Students, those of you watching this for the first time, right? Try watching the video one more time, because meiosis 1, especially prophase 1, can be overwhelming. But I have made it easy for you to understand with the help of diagrams. Very easy it is. Okay. Now understand. Now understand how that NC, 2 and 2C, how it is coming. Understand. That's all. We have a human cell. We have a simple human cell. Okay. Simple human cell. How many chromosomes? 46 chromosomes. 46 chromosomes. 46 chromosomes. Human cell, 46 chromosomes. Understood. Write down. 46 chromosomes. Okay. Now, for example, here we have a DNA which is around 40 picogram. 40 picogram DNA we have. Okay, 40 picogram DNA we have. Okay, 40 picogram DNA is there. Understand now. Now you write down a table, you write down. All of you make a table. All of you make a table like this. Understand. Okay. Now you write down here stage. Stage. Okay. Number of chromosomes. Number of chromosomes. Number of chromatids. Number of chromatids. Write down DNA content. DNA content. DNA content. Now, the main part, DNA content. Okay. Now understand. Understand now. First tell me, in G1 stage, so let me check your attendance. First, you have marked attendance or not. Friends, quickly mark your attendance. And I look so young, huh? Nice compliment. Okay. Now first stage we'll learn. First, G1 stage. G1 stage. G1 stage. Tell me the number of chromosomes. In G1 stage, how many chromosomes will be there? 46 chromosomes. Yes. G1 stage, there will be 42 chromosomes. 46 chromosomes. Yes. There will be 46 chromosomes. Number of chromatids will be double. Tell me. If there are 46 chromosomes, how many chromatids will be there? 46 only. Why? Because it looks like something like this. This is G1. This is G1. So 46 chromatid. DNA content is 40 picogram. This is your G1 stage. G1 stage. G1 stage. All of you know what happens in S phase. In S stage, what happens? In S stage, there is duplication of DNA content. This will become, there is duplication of DNA content. That is right. That is, chromosomes will be same, 46. Chromatid. Tell me what is the number of chromatid. 1 chromatid, 2 chromatid. 46 into 2 is 92. 92. Yes. DNA content 42 will become 80. 80. Beautiful. Beautiful. Understood? G2 will be same. Yes, G2 will be same. Prophase will be same. Yes, prophase also will be same. Understand? It will be same to same. Okay. Prophase, metaphase. Prophase. Right. In your G2, prophase, metaphase. G2, prophase, metaphase, and even in anaphase it won't change. Till anaphase it won't change. Even in anaphase also it won't change drastically. Same. Same. Okay, same. It will be same to same till anaphase. Same to same. In anaphase, till anaphase it is almost same to same. Now understand what happens in telophase. Understand what happens in telophase. What happens in telophase? Telophase, there are two types here. There is per cell and there is something called as per nuclei. Per nuclei. Okay. Per cell and per nuclei. Per cell will remain the same again. 46, 92 and 80 and 80. Okay. And 80. Now per nuclei what is happening? Tell me. Per nuclei what is happening? If you go back, per nuclei what is happening? Okay. We had, here we had, what do we have here? This was 2 and 2C. Here we got 2 and 4C. 2 and 4C. Right, 2 and 4C. So from 2 and 4C, from 2 and 4C, if half the number of homologous chromosomes going this side, there are totally, how many chromosomes are there? Tell me. How many chromosomes are there? Entirely there are in telophase. Tell me how many chromosomes are there? In telophase, how many chromosomes are? Tell me. In telophase, if you go, chromosome number will remain the same. 46 chromosomes are going to be there. 46 chromosomes are going to be there. There is no change in number of chromosomes here. 46 only. 46 only will be there. Okay. 46 chromosomes will be there. Okay. So can I tell, out of the 46 chromosomes, 23 are going this side and 23 are going this side. Half of the 2 and 2C, 1N is going this side, 1N is going this side. This 4C, 2C is going this side, 2C is going the other side. Do all of you understood? From 2 and 4C we end up getting N2C. N2C. If that is clear to you, all of you, I can teach you the next concept. If that is not clear, quickly ask me in the chat now. Quickly ask me. Yes? That is the 80 picogram which we had. The 80 picogram which we had, 40 is going this side and 40 is going this side. See here, per nuclei. What is happening per nuclei, students? Per nuclei. Now, how many chromosomes there per nuclei? 23 are there one side. 23 are there one side. Chromatid again. Each chromosome has 2 chromatid. Tell me, in anaphase 1, was there any separation of chromatid? No. Each chromosome has 2 chromatid. Tell me, 23 into 2 was what? 23 into 2 is 46 chromatid. Chromatid number will remain the same because there is no splitting of centromere. So 23, 46 and 40 and 40. This is at the end of meiosis 1. At the end of meiosis 1. At the end of meiosis 1. At the end of meiosis 1, a diploid cell. At the end of meiosis 1, a diploid cell did become haploid. Tell me, a diploid cell, 46 chromosomes, DNA content which is 40, okay, did become 23. Diploid is becoming haploid. 2n, 2n. So can I call this reduction division? Can I call this reduction division? This is meiosis 1. Entire thing is meiosis 1. So can I call this reduction division? Because there is reduction in ploidy. There is reduction in ploidy. Students, are you getting the deeper concept of meiosis today? This is how you should learn. Okay? Understood? Now students, from here meiosis 2 starts. From here meiosis 2 starts. Meiosis 2. Meiosis 2 starts. Again. Prophase we have. We have prophase. Prophase 2. This is all 1. This is all 1. Okay, this is all 1. Prophase 2. Yes, prophase 2. Will there be any change? No, there will no change. 23, 46 and 40 and 40. Okay, 40. Now, even prophase 2, even metaphase 2, it will be same to same. It will be same to same. Now, imagine, not imagine, I have told you in anaphase what happens. I have told you in anaphase of your mitosis what happens, tell me. In anaphase what happens? There is splitting of centromere in anaphase of mitosis. And I have also told you the anaphase 2 is similar to mitosis. Tell me, yes or no? Anaphase 2 is similar to mitosis anaphase. There will be splitting of centromere, and each centromere, so each splitting of centromere, and each chromatid will act like a daughter chromosome to the future nuclei. Students, this concept I have told you in your mitosis. Each chromosome will act like each chromatid will act like a chromosome. Here, each chromatid will act like a chromosome. Will act like a chromosome. Yes, will act like a chromosome. I've told you this. In the case of mitosis, is this anaphase 2 just like your mitosis? Yes, it is just like mitosis. Now, each chromatid is acting like a chromosome. So, can I tell this 46 chromatid will act like 46 chromosomes now? Yes, 46 chromosome. Yes, 46 chromosome. Number of chromatid will be what? Same. Because we only have one chromatid now. Each chromosome has only one chromatid. So 46, 46. Change in DNA content? No, there is no change in DNA content. 40. No change in DNA content. Now understand. Now finally we come to telophase. Telophase again we learn in the terms of cell or per nucleus or nuclei or per nuclei. Per cell will be same. 46, 46 and 40. Now understand, let's go back here. What is happening here, students? I told you, tell me in this telophase, totally how many chromosomes are there now? 46 chromosomes are there. Right, 46 chromosomes are there. 46 chromosomes. 46 chromosomes. 46 chromosomes with one chromatid. Why? Because as a result of anaphase, 46 chromosomes with a single chromatid. Now, out of this 46 chromosome, can I tell 23 are going this side and 23 are coming this side? Can I tell? In telophase 2, per nuclei, 23 are going top, 23 coming down. Understanding? Yes, 23. So, what is happening per nuclei? Per nuclei, how many chromosomes then? Per nuclei, there are 23 chromosomes now. 23 chromosomes. How many chromatid? Everyone is single chromatid. Single chromatid. So, 23. 23. Now, students, understand. DNA, half of the DNA content is going this side. Half of the DNA content is coming this side. If there was 40 DNA content was left, 20 will go this side and 20 will go this side. Yes. 20 will go this side. So how much is left now? Per nuclei there is only 20 left. 20 left per nuclei. 20 left. 20 left. 20 is only left. Not 40. Not 40. Only 20 is left, can I tell? 20 is left now. Per nuclei it is dividing. Per nuclei it is dividing. 20. Now students, if 20 is left here, can I call this NC now? Because here it was 2C. Understand? Here students, it was N2C. Here, by the end of telophase 2, per nuclei, it was N2C here. N2C. It was N2C. Right, N2C. Now this C is also splitting up. DNA content is splitting up. So can I tell this is becoming NC now? It's becoming NC. Oof. This is called as reduction division. Reduction division. From one diploid cell, one diploid germ cell, we are getting 4 haploid gametes. 4 haploid gametes. How many of you followed? Tell me. How many of you followed till now? If you did not follow, please watch it again. You write it down and learn it. Because I'll show you proof that question can come from here. Beautiful, right? This is how meiosis works. Tell me how many of you learned meiosis this way. Tell me how many of you learned meiosis this way before. Such an easy concept. Okay. This is the beauty of chroma. Now you will ask me, sir, we started off with 40. We started off with 40 picogram. And you're telling me from one DNA. So you're telling me from 40 picogram, we are getting 24 haploid. 4 haploid cells. How is it possible? How is it possible? From 40 doesn't make sense. Friends, remember, in the middle we have something called as S phase. In the middle we have something called as S phase. In the S phase, what happens? The DNA content doubles in S phase. So this 40 became 80 and 80 was divided into 20, 20, 20 and 20. Understood? Duplication of DNA content. And only happened only once. Line of your NCERT. It happened only once. Understood all of you meiosis? This is your chart for your meiosis. That's all. Done. Easy, right? Yes, it is easy. It may look complicated because you might be hearing for the first time. Okay. You might be learning like this for the first time. So I would advise you to sit and learn it one more time. Go through the session one more time. Any doubt, anywhere, you can always contact me on the Telegram channel. Right? That is Telegram right here. Telegram. All of these notes will also be available on the Telegram. Okay? Telegram channel. Done. Now students, after a whole brain exercise, I feel some amount of DNA content in your brain also is reduced. Some cells has been consumed completely in your life. Yes, it's good. Now what we'll do is, yes, ask the doubts. What we'll do is right now, we will take a small 20-minute break. Okay. So I can rest my voice. All if you can refresh. After 20 minutes break, can we solve some PYQs, some numericals? and some amazing questions to solidify our information which we learned today. That is, whatever information which we learned today, the application of it. Application of it. Yes, application of it. And if you look at the questions, can you see? I have so many questions for you. Can you see? So many questions for you today. More than 25 questions are there. More than 25 questions are there. So you can solidify your information. Okay? You can solidify your information today. And revision. Okay? And revision also happen by side by side. Okay? So what is the time right now, students? 7:36. 7:56. 20 minutes break is required. You just have to sit and relax for some time. Okay? So we'll resume the class at 7:56. Resume the class at resume at 7:56. Okay? 7:56. 36, 46, 56. Okay? 20 minutes break. Chill and come for some time. Drink some water. Eat some food. Make your brain recharged. Exactly at 7:56, I will see you. Okay? 7:56 p.m. After that, we'll solve questions. Okay? Now, scoot, everyone. Run away. Smash the like button, run away. Quickly, run away. Yo, welcome back to the channel. Welcome back. Okay. Question Krishna Veeni. Okay, Krishna, let me tell you. If you attend today's class, you would have learnt about meiosis. Now try solving the questions which are NEET PYQ and see if whatever I have taught, is it actually applicable for you. It's your call. Don't listen to anyone. Don't listen to anyone that this channel will be helpful or not. Always listen to yourself. That is, you learn the concept here, try solving the PYQs. If you're not able to solve PYQs, it means you have not learned properly. If you're able to solve all the questions, it means you have learned properly. That's all. That's all, is it? There is no rocket science here. Now let's see the question here. Very simple question. And if you have learned today's class, you will be able to solve this question. Simple as it is. And this message is for all of you students. This message is for all of you. We always prioritize your studies, and it is always best for you. Know what is best for you, right? And if you think that whatever we are teaching here and it is helpful to you, we always believe that yes, it will be helpful to you. Try solving these questions, you will know on your own. Try solving this question. Okay, Krishna, try solving this question. If you're able to solve this question, yes, you can clear NEET. If you're able to solve the questions which I am teaching you, because all of these are NEET PYQs, NEET 2024 PYQ, NEET 2022 PYQ, all the PYQs are here. If you're able to solve these questions, you're good to go. Okay, see this question now. Match the following. They've given you 4 options here. All you have to do is match the following. First of all, tell me. First one, diakinesis. What happens? Last stage. Yes, diakinesis is the last stage, right? Yes, diakinesis last stage. What happens in diakinesis? Yes, in diakinesis I told you there is termination of chiasmata. Have I told you or not today? Tell me. In diakinesis there is termination of chiasmata. So A is gonna be 2. Tell me, if A is 2, how many options are there? Tell me how many options are there? A is 2. Only one option is there. Yes, only one option is there. A is 2. So you don't even need to read the other options, you can easily go. Now B is 4, Packettine stage. Tell me, appearance of recombination nodules. Apart from recombination nodules, what else happens in your Packettine? Tell me, one by one. Crossing over, recombination nodules. Yes, crossing over takes place, recombination, bivalent tetride are most clearly visible here. Yes, apart from that, there is also an enzyme called as recombinase enzyme. All the following points you should remember about Packettine. Crossing over, recombination nodules, then bivalent tetride is easily visible, and last one is going to be your recombinase enzyme, all of them. Now C is going to be one, that is zygotine, formation of synaptonemal complex. All of you remember synapses, formation of synaptonemal complex, and also formation of bivalent and tetrad. That's all. Leptotine stage. What is leptotine? D is going to be 3, that is chromosome look like thread. Starting stage, right? Starting stage. So each and every part of every single stage, you should know what is happening one by one. Next question. See here. Chromosome become gradually visible under the light microscope during leptotine stage. I told you when I was teaching you this, right? The beginning of diplotin stage is recognized by dissolution of synaptonemal complex. Tell me. D for diplotin, D for dissolution. I told you. Dissolution, diplotin. Obviously, both the statements are correct now. Yes. Both statements are correct. Easy. Don't you think so? This NEET 2024 PYQ was so easy. If you just read the NCERT line by line, understand every single point, it becomes so easy now. So easy. Yes, so easy. Look at this next question again. Packettine, zygotine, diplotin, dileptotine. Again, match the following. We just now learnt about leptotine, that is visible under the, where is it? Chromosome become gradually visible under light microscope and conversation continues. Yes. B is going to be 4. Yes. What is chromosome starts pairing together synaptonemal complex? That was your one second. Let's go one by one. First of all, chromosome starts pairing together to form synaptonemal complex. That was the zygotine. A is 2. Yes, A is 2. Chromosome info, that is leptotin, I told you. Chromosomes of bivalent chromosome becomes distinct and recombination nodules, I told you just now, is packaging. Yes, C is 1. And dissolution of the complex, diplotin. Tell me what is the answer now. Tell me the answer. 4 1. Tell me, is it difficult or easy now? Tell me. Difficult or easy? We have learned every single point. We have learned every single point. Every single term you learn about packaging, what should come to your mind? Dissolution, packaging, crossing over, recombination nodules. Yes, recombinase enzyme. As well as tetrad is clearly visible. That was given here. See, it is clearly visible now. That was the point here. Zygotin, what should come to your mind? Zygotin, what should come to your mind? Formation of synaptonemal complex, synapses, formation of diad tetrad. Diplotin, dissolution of synaptonemal complex. Leptotin, gradually under microscope. Everything. Next question. The process of appearance of recombination nodules occurs at which stage? See, NEET 2023 question. Recombination nodules, what should come to your mind? Recombination nodules, packaging. Recombination nodules, packaging should come to your mind. Packaging. NEET PYQ. Next question. Which of the following stages meiosis involves division of centromere? Division of centromere. Now, division happens only in anaphase. But in anaphase 1, division of homologous chromosomes. Anaphase 2, centromere divides. Anaphase 2, centromeres divides. Anaphase 2, centromeres divides. Why? Because anaphase 2 is just like your anaphase of mitosis. Centromere splits. We know that. We know that. Option number 4. Anaphase 2. Next question. See. Tetrad, state the following correct option. Correct option. You have to find correct option. A very easy question again. Tetrad formation is seen during leptotin. Students, tell me. What happens in leptotin? Chromosomes become gradually microscope. So no. During anaphase, the centromere splits and the chromatids separate. Yes. They have not mentioned anaphase 1 or 2. So this statement is from mitosis now. This statement is from mitosis. Yes. Statement from mitosis. Correct. Okay. Termination takes during packaging? No. Termination of chiasmata happens where? At the diakinesis, not packaging. Yes. Nucleolus, Golgi complex and ER are reformed during telophase. Yes. They reformed during telophase. Crossing over, correct. Okay. Correct. Telophase, they come back. Crossing over takes place during sister chromatids homologous chromosomes? No. Why wrong? Because it happens between non-sister chromatids. Non-sister, not sister. Non-sister. So the statement is also wrong. So what is the answer now? What is the answer? B and D. Option number 3. Option number 3. Again, very easy concept. All of you are able to answer because you know it now. Today you learned meiosis. You have mastered meiosis today. Okay. Let me know in the comment section today how was meiosis today. Okay. Next question you see. The dissolution of synaptonemal complex occurs where? I've told you 100 of times already. Dissolution is diplotin. Is diplotin. Diakinesis termination, packaging, crossing over, recombination nodules. Leptotin, gradual visual under microscope. It's a NEET PYQ from 23. NEET PYQ. Next question. During which stage of mitosis and meiosis, respectively, does the centromere of each split? In mitosis, where does it happen? Mitosis, anaphase. Meiosis, anaphase 2. Anaphase 2. Anaphase, anaphase 2. Splitting of centromere. We learned it today only. So students, there are only few questions we can argue from cell chapter. And if you understand the meiosis and mitosis from my class, there is not even a single question which you can't answer. Okay? This question now. Select the correct sequence of substages of prophase 1. Correct sequence: leptotin, zygotin, packaging, diplotin, diakinesis. Let zebra play during the day. Let zebra play during the day. Done. Do the match the following. That's all. Very easy answer again. Let zebra play during the day. Look at this question now. Which one of the following never occurs during mitotic cell division? Okay, it's a mitotic question. Okay. Calling and indecision chromatid happens. Spindle fibrillary to kinetochore, metaphase, yes. Movement of centrioles towards the opposite poles, yes, it happens in the prophase. Pairing of homologous chromosomes does not happen, because pairing of homologous chromosomes happens in the case of meiosis. It happens in the case of meiosis, not in mitosis. So 4 is the option. Never happens. Never happens. Next question. The appearance of recombination nodules on the homologous chromosomes during which phase? Again, homologous chromosomes. That is, appearance of recombination nodules on homologous chromosomes during meiosis characterizes. What happens? At Pachytene, crossing over happens. At Pachytene, crossing over happens. Crossing over takes place, Pachytene. Now, regarding meiosis, which of the following statement is incorrect? Incorrect regarding meiosis. 4 haploid cells are formed at the end of meiosis 2. 4 haploid cells. There is 2 stages, meiosis 1 and meiosis 1 and meiosis 2. DNA replicates occurs in S phase of meiosis 2. Pairing of homologous chromosomes and recombination occurs in meiosis 1. Incorrect statement. I'll give you 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Students, tell me, DNA replication happens only once. That is an interphase, and it is before meiosis 1. Does the DNA replication happens in interkinase? No. That is, DNA replicates once in S phase in meiosis 2, does not happen. In meiosis 2 does not happen, right? Simple answer. 3 is the answer. Simple answer. Now, in prophase 1 of meiosis, chromosome starts pairing together and synapsis takes place. This process occurs during which phase of stack? Again, NEET 2022 question. Same question repeats every year. The same pattern of questions. Tell me, synapsis, zygotine, synapsis, formation, synaptonemal complex. Next question. The beginning of diplotin is characterized, by dissolution of synaptonemal complex. Yes, diplotin, dissolution of synaptonemal complex. Okay, dissolution of synaptonemal complex and formation of X shape called as chiasmata. Yes, chiasmata formation takes place. In oocyte of some vertebrates, diplotin can last for months or years. I told you, months or years it can last. Both are correct. But is it explaining this? No, it is not explaining. So option number 4, not correct explanation of A. Not correct explanation of A. Again, easy question. Next one. Oocyte of some vertebrates gets arrested for years of months in which stage? At diplotin stage, we just now learned. Same question, different format. Same year, students, same year. NEET 2022, same question, different format. Diplotin. At diplotin stage. Next question. Select the correct sequence of events occurring at the prophase 1 of meiosis 1. Again, you need to think and answer because many students have gone wrong here. 40% of students cannot answer this. Okay, see this. Tell me what happens first. Compaction of chromosomes. Yes, compaction of chromosomes. Formation of synaptonemal complex, zygotine. See, this is in leptotene, zygotine. Okay, Pachytene. Then we have diplotin is not there. Diplotin is not there here. Diplotin is not there. Termination of chiasmata, that is diakinesis. Now, did I tell you at the end of diakinesis, that is late prophase, one line of NCERT I showed you, breakdown of envelope. This is the last. So what is the answer? C, B, E, DNA, cbDNA. Yes, students, I know you might have gone wrong here because it was a little complicated. You might have thought breakdown happens first. You might have thought. Let me show you why you're wrong. I think I showed you also when I was teaching you this. When I was teaching you the last part of your prophase 1. At diplotin, diakinesis, I showed you this. At diakinesis, I think I showed you this. Here. Nuclear envelope disappears after diakinesis. Did you understand? After diakinesis. That's what I told you. End of prophase 1, late prophase. Understood? So you can make such mistakes. It's only a matter of revision. It's only a matter of revision. If you revise, you will never make mistake. Don't worry. You're good to go. Okay. How many students do you realize? So many questions we did in this span of time. So all of you understood this question now? Understood this question? First one is going to be your leptotene, right? Leptotene. This is zygotine. This is going. Then we have pacatine. Then we have diakinesis. This is the end. Let us late prophase. Late prophase. That's all. Late prophase. Simple, right? Next question you see. In meiosis, crossing over and exchange of genetic material between homologous chromosomes are catalyzed by an enzyme at pacatine stage. The enzyme is called as recombinase. Very simple. Recombinase. Next question. Which stage of meiotic prophase show termination of chiasmata distant future? NEET 2021 question. NEET 2021 question. Tell me, where does it happen? That is termination of chiasmata, diakinase. You cannot go wrong anymore. You cannot go wrong at all. Direct questions come from here. Okay, diakinesis. Termination of chiasmata. I told you, chiasmata terminates. Termination of chiasmata. Diakinesis. Next question. It's a good question. Now, students, you might think, sir, why did you teach the DNA content? That was a little complicated, no? DNA content was a little complicated. You might ask me, sir, why did I teach you DNA content? Now you'll see the result. Why? I teach you the DNA content. Okay, see this. After meiosis 1, the resultant daughter cell will have. Same amount of DNA as in the parent cell in S, twice the amount of DNA in the comparison to the haploid, same amount of DNA in comparison to the haploid gamete, 4 times the amount of DNA in comparison to the haploid gamete. Go. Time starts now. After meiosis 1, the resultant daughter cell will have. After meiosis 1. Time starts now. Go. After meiosis 1. Now, students, all of you who are going wrong, I'll tell you why going wrong. Now you tell us. Now, we'll go back now. After meiosis 1, the daughter cell will have. We'll check the first option. This is slide number 71, right? Slide number 71. Okay. Same amount of DNA as in parent cell. After meiosis 1. After meiosis 1. Students, tell me. After meiosis 1. This is the end of meiosis 1. End of meiosis 1. Okay. It is N2C. Does it have the same DNA content as the parent or the S stage? Here it is 2N, here it is N. So first option is wrong. This is slide number 33. Okay. Okay. No, wrong. Twice the amount of DNA in comparison to haploid. Twice the amount of DNA. Does it have twice the amount of DNA? Twice the amount of DNA compared to the haploid. Compared to the haploid gamete. Twice the amount of DNA. Here we have DNA content. They are asking DNA content. Here we have 2C. Here we have C. Is the DNA content double or yes or no? You look at this and tell me. Compare after meiosis 1, does the daughter cell have twice the DNA content compared to the haploid gamete? Yes or no? You tell me now. Yes or no in the chat. That was the second option. Okay. That was the second option. Okay. Now, same amount of DNA in comparison to the haploid gamete. No, haploid gamete was N. After meiosis, it was NC. It was 2NC. See, second one is same amount of DNA in comparison to the haploid gamete. Here we have N, here we have 2C, we have C here. So the DNA content is obviously not the same. DNA content is obviously not the same. So third option is also wrong. Fourth option is also wrong because it says 4 times. Fourth option is also wrong because it says 4 times. So what is the answer then? Twice the amount of DNA. Amount of DNA is DNA content. DNA content. Here we have 2C, here we have C. Okay. So this is the right answer. This is the right answer. Students, this is the reason you need to know the table. You need to know the concept. For you to answer such questions from NEET 2019. NEET 2019 question is this. NEET 2019 question is this. Good question, right? Tell me. Very good question, right? So it goes on to show you that yes, whatever I'm teaching, if you apply it, you will be able to solve all the questions. There are some out of NCERT questions which have been asked, but those questions, they will come in option part B, where you can keep a multiple option. Okay, understood this question. Look at this next question now. Are you able to see the question, students? Are you able to see the question? A fruit fly has 8 chromosomes. So 2n is 8 chromosomes in each cell. In each cell, 8 chromosomes in each cell. During interphase of mitosis, the number of chromosomes at G1, is 8. Okay. What would be the number of chromosomes after S? After S. At S, this is a 2n. At S, what is the number of chromosomes at S? What is the answer? Tell me. What is the at S? At S, what is the number of chromosomes? Concept question. Concept question. Very simple, very easy. Very simple, very easy. Can I tell you? The answer is very simple, students. In mitosis, is there change of number of chromosomes? You tell me now. In mitosis, when I was teaching you mitosis, in mitosis, is there any number of chromosomes? The question is asking you number of chromosomes, not DNA content. Number of chromosomes. If the number of chromosomes in mitosis, will it always remain the same? From top to bottom in the cycle, will remain the same, right? Number of chromosomes will remain the same. Number of DNA content can vary. Right? Do you remember I told you? When I was teaching you mitosis, I told you the DNA number of chromosomes never vary. Yes, only the DNA content varies. Remember? You don't remember, huh? One second, let me show you. I think I've told you multiple times. I've told you the DNA content can double 2n 2c to 4c. So if we get 2n 2c, it becomes 2n 4c. But what is the number of chromosomes? Number of chromosomes will remain the same. So if it was 8 here, it will also remain as 8 here. Simple. So answer is 8. Answer is 8. So students, you will get such questions which will trick you little. And will make you think and all. But you should remember the basic concept of every single topic. Okay. Now since you have encountered such questions, will you ever make a mistake in such questions now? No. You will always think now. You will always think. Okay. Look at this next question now. If there are 20 centromeres at anaphase, if there are 20 centromeres at anaphase, how many chromosomes are there in each daughter cell following cytokinesis? So let's draw anaphase here. very easy question. Only you know the concept also. You know the concept for this. All of you know the concept. Okay, all of you know the concept also. All of you know the concept also now. Okay, understand. Here at the anaphase, there are how many? There are 20 chromosomes, 20 centromeres. 20 centromere. Here the meaning of 20 centromere, students, the meaning of 20 centromere is 20 chromosomes. Yes. 20 centromeres means 20 chromosomes. 20 chromosomes. So in the anaphase, what happens? In the case of anaphase, out of the 20 chromosomes, can I tell 10 is going this side and 10 is going this side? Yes, and it is splitting up. It is splitting up. It is splitting up here. Tell me, if the 20 centromeres are there, it is not 20 chromatids. It is not 20 chromatids. It is 20 centromere, means 20 chromosomes. So always remember, the number of centromeres resembles the chromosome. How many centromeres are there? How many same number of chromosomes will be there. Okay. So 20 is going there, 10 is going there. So what is the answer now? So 10 after cytokinesis. What will happen? 10. Students, all of you know the mistake you're commenting. All of you know the I saw the chat. Don't worry. All of you know the mistakes which you which you did. Don't make the same mistake again. Now, students, tell me, are the numericals a little tricky, in mitosis also? Tell me, are the numericals a little tricky here? Yes, right? So what we'll do is how we did for genetics. How we did for genetics, a proper numerical class, when the exam is coming near, not now because you'll forget, I know. As the exam is coming near, I will keep a proper numerical class for entire biology. What do you say? What do you say? For entire biology, one numerical class. That will come up towards the end of the session. So if you want that, let me know in the comment section today also. And smash that like button, subscribe to the channel also. Okay? Smash that like button. Look at this question now. Meiosis has evolutionary significance because it results in, obviously, it results in 4 haploid cells. Yes, it results in 4 haploid cells, egg and sperm. But what is the main reason? What is the main reason that it is giving us evolution? I told you, evolution is because of variation. Evolution is because of variation. And variation is because of recombination. Variation is because of recombination. Remember, students, it can become very tricky. See, you know this is also right. You know this is also right. But when you get such questions, you should always go for most probable answer. The question is asking you evolutionary significance. Evolutionary significance. And I've told you, as a course of evolution, if evolution has to take place, variation should happen. Variation needs to happen. And how do we get variation? Because of recombination. That is crossing over. That is crossing over. Understood, all of you? Understood. So such questions are a little tricky sometimes. You need to identify those questions. Okay. Deliberately I've added the tricky questions till the very end. So for you to realize, yes, there will be easy questions. Yes, there will be easy questions. But there will be few tricky questions which needs practice so that you don't become overconfident. Okay. Now this is your homework question now. This is your No, this is not homework question. This is not homework question. Understand? Now, a bivalent has. What does bivalent has? Tell me. What does bivalent has? What does bivalent has? 2 chromatids and 1 centromere. 2 chromatids and 2 centromere. 4 chromatids and 2 centromere. 4 chromatids and 4 centromeres. Tell me, go in the chat. This is not homework question. Do this question right now. Do this question right now, all of you. What is a bivalent? Bivalent. If you remember the structure of bivalent, this is how I drew it. This is your bivalent, or also called as tetrad. In the between we have synaptonemal complex. So how many chromatids are there? How many chromatids do you find here, students? How many chromatids? 1, 2, 3, 4. 4 chromatids. How many centromere? 1, 2. 2 centromere. This is your homework question. No, 1 second. Let me just check. No, we have more questions. Oh my God, we have more questions. We have more questions. Okay, not homework question. This is I have 2 more questions. Meiosis 1 is followed by. It is always followed by interphase. Meiosis is followed by any of the period of the rest. Is followed by period of interkinase. Is sometimes followed by an interphase. Tell me, what is the answer? This is easy question again. So after your meiosis 1 diagram which I drew, after meiosis 1 do we have interkinase, then meiosis 2, right? Interkinase and then meiosis 2. Yes. So after your meiosis 1, interkinase, meiosis 2. Nice. Now, bivalent chromosomes can clearly appear as tetrad during when? Question is very simple. Question is very simple. Bivalent chromosomes clearly appear as tetrad during. Go. Let me drink some water from my pink bottle. Students, smash the like button, all of you. Quickly smash the like button. I've told you formation happens at the zygote. Formation happens zygote. Clearly visible at pachytene. Pachytene. And students, remember, almost 40% of the students could not answer this question correctly because they clicked on zygote. They clicked on zygote. Okay. This is your homework question. This is your homework question. In the comment section, I want homework question in the comments. In the comments. And also let me know in the chat. Also let me know in the chat today's video, how was meiosis, students? How was meiosis? How did you enjoy meiosis, right? And I believe it was first time all of you learning the entire NC 2 and 2C. And first time you have understood the meiosis in a conceptual level. First time you understood meiosis in a much more conceptual level. So let me know in the comment section, how was the meiosis? I'll be checking, all of you. And all the new students also, please smash that like button. Okay, I'll be checking. Okay, the students, you need to realize that only learning all this concept is very easy. But how do you get more clarity on this? The more clarity will come by you solve more questions. Only when you solve more questions, more clarity comes in. Students, I've been telling this very long time. I've been keep on telling you. Please join a test series. Please join a test series, which will actually help you. And don't wait until December, January, February, March, or April to join a test series. That's my honest thing. Please join test series as soon as possible. It's fine if you spend some money because this is an investment, right? So join a test series, good one, which can help you to solve all these questions. Join a test series as soon as possible. Okay. So with that being said, students, let me know in the comment section how was today's class because I will be reading the comments religiously. Okay. Don't later on don't tell me, sir, I'm very lazy to type. Go and type. Okay. So bye bye, students. Take care. Have a great day ahead. Okay. And keep on studying. And the next class in cell line by line. Next class in line by line, I'll be taking photosynthesis in higher plants. Photosynthesis in higher plants. Next chapter on line by line. Okay. Initially, I thought I'll do biomolecules, but let's take a small gap from cell. Let's go to photosynthesis in higher plants. We'll finish photosynthesis in higher plants, then we'll come to biomolecules later on. Okay. So bye bye, students. Take care. And all the DNA students in the chat, I'll see you tomorrow in DNA batch. Okay. Tomorrow we'll be doing anatomy of flowering plants in detail. All the students in DNA batch. Okay. So take care, all of you. Bye bye.